Turns out that projector issue was not just me, that's campus-wide. Okay, good morning, everyone. Today, you'll be pleased to hear, is the last set of new content for this course. So you can stop remembering things after today. Today, we are going to be talking about one of my favorite areas of research, which is coevolution, which is building on some of the stuff we've been looking at recently with evolutionary epidemiology. Before I get started, a couple of announcements. Just a reminder today, homework 4 is due. A week today, your projects are due. And then for those who are graduate students in Math 795, I will send out an email or an announcement about arranging a time slot for you guys to do your presentations. So there'll be a 10-minute presentations with a little bit of Q&A after that. These will most likely be the following Tuesday or Wednesday, which I believe is the 6th or 7th of August. So I'll try and find a time that's mutually convenient for everyone who's in Math 795. Okay, I think those are all. Oh, the other major announcement is, yeah, so Monday, just a reminder that there's no class on Monday. Please use this time wisely to finish up your projects if you haven't already. If you, for any reason, are unable to submit your project on time, please let me know. There are penalties for it being late. It is quite a large component of the course. Okay. Any questions before I get started on today's material? No? Okay. Okay, so coevolution. So for the last however many weeks, think back to how young you felt, I don't know, 3 months ago, whenever it was, beginning of May, when we started, way back. And since then, we've been focused on evolution of, say, a single trait or a single allele, perhaps, or at least a single genetic locus or a single species. We have done like multi-locus models, but those would still, say, be coming together perhaps to form a particular phenotype. We haven't been thinking about necessarily multiple phenotypes. You do it a little bit in that in that population genetics framework. We haven't been thinking about coevolution of things though. And I'm really going to focus on coevolution in across species. You can have essentially coevolution of traits within a species, but we're not going to talk about that today. So focusing on evolution of a single species, especially in our adaptive dynamics framework, is probably the easiest way to think about this. You know, we've had one trait that's evolving, and we've been able to derive things about, you know, what's going to happen to that trait in the long term. You know, it's going to, you know, our selection gradient tells us how that trait is going to evolve, which direction is it increasing or decreasing. We eventually reach, say, a singular strategy or a boundary. But if we're at a singular strategy, then we can say, look at our higher order derivatives will tell us something about, first of all, do we converge to that singular strategy, but also is that singular strategy evolutionarily stable? In other words, is this like a fitness maximum? Is it uninvadable? Okay, so we've focused on these questions and we've covered with single species. Essentially, we've looked at resource competition, we've looked at pathogen evolution, so how does virulence evolve or drug resistance, and we've also looked a little bit about host evolution as well. The final bit of the course, which is just today, is going to be talking about this idea of coevolution between two species. I'm going to focus on two species because this is the simplest case. We can have coevolution between many species. So the key thing about coevolution is that, which I'll get onto in a moment, is it's all about this idea of reciprocal adaptations. Okay, so rather than say there being a constant environment, your environment is now changing. We'll talk a little bit more about that in a moment. So today, we're going to cover this idea of coevolution. I'll talk about the difference between a biotic and an abiotic environment. Talk about something called Red Queen dynamics. It's a particular way of thinking about coevolution. We're going to extend this idea of a singular strategy to what happens if we have two traits evolving, two species evolving, so we have co-singular strategies. Talk a little bit as well about mutation supply and also coevolutionary cycling at the end. Okay, so by the end of today, you should be able to understand the structure of and analyze some simple models of coevolution. We're also going to dip back into population genetics right at the end to come full circle. Okay, so this is quite a wordy slide, but I just wanted to give you guys some background about coevolution and why I think it's interesting. So, you know, if we're just thinking about, say, one species evolving, we might just be thinking about it adapting to some constant environment. That environment might fluctuate. You might think of environment typically as just being, what is the temperature, what, resources are available, what's the pH, any of those kinds of things. We refer to those sorts of things typically as the abiotic environment. Okay, so non-living aspects. But they'll also be adapting to biotic or living aspects of their environment. So you can think of a biotic environment as everything else that it's interacting with that is living. And the key difference between a biotic and an abiotic environment is that, well, biotic environments are themselves going to be subject to natural selection, genetic drift, other processes of evolution. Okay, so those biotic environments themselves can evolve. And if those biotic environments evolve, we would expect there to be a feedback loop, right? So if we think about antagonistic interactions between hosts and pathogens or predators and prey, we might think of pathogens adapting to their hosts and then hosts adapting to their pathogens and vice versa. And this constant changing environment means that it can lead to quite really interesting dynamics and helps us to understand a wide range of biological phenomena. So to sort of properly define coevolution, it's a process of reciprocal adaptations between two or more species. This was first discussed by Charles Darwin all the way back in The Origin of Species, thinking about the context of not antagonistic interactions, but cooperative or mutualistic interactions, so between flowering plants and insects in particular. And then one of the sort of really interesting early ideas about coevolution was this identification of this orchid here that had this really long spur. So the nectar for this orchid is stored all the way at the bottom. But at the time, there wasn't any insects or any pollinators known that were able to reach this. But it was hypothesized, well, there must be something that has co-evolved with this particular orchid that would have a long proboscis. So later on, this hawk moth that is able to feed on the nectar and hence pollinate plants with these very long spurs was identified. So thinking about coevolution has helped to understand some of the patterns and different variety that we see in nature. I mainly think about antagonistic coevolution. For me, this is the most interesting area because antagonistic relationships can lead to very rapid adaptation, can help us explain lots of different biological phenomena. So antagonistic, basically thinking there's a victim and an exploiter. So host and pathogen, predator and prey, that kind of scenario. And it can lead to this very rapid selection for adaptations and counter-adaptations. So the idea of antagonistic coevolution has been implicated in lots and lots of different biological phenomena, things that you might not ordinarily think would perhaps be connected to, say, infectious diseases. So one of these, evolution of diverse immune responses. So we've been talking about the evolution of host immunity in the last lecture. We have innate and adaptive immune responses. There's a wide range of different immune responses, even in microbial populations. So you can experimentally evolve and co-evolve, say, bacteria and their viruses in the lab, and you can see that they end up with different immune responses depending on different conditions. You can also see that if you just say evolve your bacteria with a virus that is constant, so it's not evolving, you just keep replenishing it, then evolution proceeds at a certain pace, certain amount of adaptation of resistance in your bacterial population. But then if you co-evolve them together, you see this explosion and really rapid acceleration of evolution, coevolution in this case. So coevolution can really sort of drive a lot of molecular and as well as phenotypic changes. Things like peacock's tail and other bright displays or sexually selected traits. These are known as secondary sex ornaments, to advertise, that you're a potentially good mate. We see this throughout nature. And these can signal mate quality. Big question is, why would these traits evolve? One of the reasons that these traits might evolve is to indicate that you are perhaps either currently not infected and therefore a low-risk partner, or that you just have good genes that confer resistance to disease. And if you mate with this individual, then you can pass those good resistant genes onto your offspring. So there are lots of these theories that have kind of sort of as to why these traits might have evolved, all about mate choice, really, and sexual selection. one of the big problems with this is how do you keep this process going? So say females choose the highest quality males and get all of those resistance genes into the next generation, into their offspring. Well, what happens in the next generation? There's very little variation. Now, this is actually known as something called the Leck paradox. Because say females choose amongst males and they pick the highest quality mates, that erodes the genetic variation that you have in future generations. So what would keep this process going? Well, for example, if your pathogens are constantly adapting to your hosts, then what might be good resistance genes now are not necessarily good resistance genes in future. And so it can keep this process going. If you just had one-sided adaptation, it can stall. Coevolution can keep some of these processes that we see going. The evolution of sexual reproduction as well. Thinking more broadly about modes of reproduction. Why does sex exist as a mode of reproduction when asexual reproduction, just cloning yourself, would be way more efficient? This actually came to be known as something called the Red Queen hypothesis. We'll talk a bit more about the Red Queen in a moment. So the idea here is that, if you have a population of males and females, and just for argument's sake, they're going to say roughly equal males and females. We've talked about equal sex ratios before. And then you've got, you can think of it as a mutation, perhaps, where a female can now just clone herself. She doesn't need to produce males, and she just produces clones of herself, and those clones can copy themselves and so on. So all of her offspring are going to be asexual females that don't need males around, they can just clone themselves. Now, all else being equal, that population should grow at twice the rate of the sexual population. So that mutation should spread. It's twice as fit, right?
Does that lead to the same issue as that you just mentioned, where there's lack of variation then?
That's the question then. So why would we be maintaining this variation? So people say, like, oh, sex is good because it generates variation, but why do we want that variation? So then we need, yeah?
Oh, I was going to say, isn't it so that you're more resistant to disease?
Exactly. So this is what people exactly thought, like, well, what is the process that could maintain this? An idea would be a coevolving antagonist, like a pathogen. So if you need to, you can think about producing diverse offspring. Some of them will be resistant to disease. Some of them won't be resistant to disease because you're shuffling all your genes with other individuals to produce your offspring rather than producing a homogeneous clone, if you like. That can sort of lead to higher average fitness. This has led to lots and lots of theory as well as experimental tests to try to understand, okay, well, under what conditions do we expect this to actually work? So I like coevolution, I like infectious disease research because it doesn't just talk about, public health, which is very interesting and important by itself, but it also tells us about lots of these patterns that we see in nature as well, ways you might not have thought about before. In general, we might expect coevolution to just help generate diversity and also maintain it. Diversity at the sort of genetic level, but also at this sort of phenotypic level as well. We'll talk a bit more about that as well later. So very good intuition before about, this idea of, infectious diseases might help to maintain sex. This became known as the Red Queen hypothesis. And more generally, we can sometimes talk about coevolutionary dynamics with this analogy of the Red Queen. So I hope everyone's familiar with Alice in Wonderland. Yeah, okay, good. There's another book you might be less familiar with that Lewis Carroll wrote called Through the Looking Glass. Who's read Through the Looking Glass? One person, yes, great. Okay, so in Through the Looking Glass, Alice meets the Red Queen and they start running. And Alice is looking around and she's running as fast as she can and she's not moving anywhere. And she says, basically, why aren't we going anywhere? And the Red Queen says to Alice, well, you see, it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run twice as fast as that. So what does that have to do with coevolution? Well, this is often taken as an analogy for coevolution, with this running being essentially adaptation. So you're having to run as fast as you can. In other words, you're having to adapt continuously as much as you can. This idea of a same place is really all about relative fitness. Sorry, my pen's gone a bit crazy. It's all about relative fitness. So running as fast as you can just to maintain some level of fitness basically says, if you stop running, you're going to fall behind. But you can never outrun your antagonists, or rarely outrun your antagonists. Okay, so this idea of the same place is about your relative fitness. You may be adapting as fast as you can. I say you adapting as fast as you can as a sort of population is adapting very rapidly to its antagonist, but it can't necessarily ever escape it through adaptation. Okay. This isn't always the case, obviously. There might be cases where it does escape, but this can help us to understand this coevolutionary process and why, these battles potentially seem to go on forever. Okay. So the Red Queen is often invoked as an explanation or an analogy for coevolution. Yes.
Could we think of this somewhere else as being higher fitness?
Yes. Yeah. So if you want to get to, one other way of thinking about this is that you're on this fitness landscape. We've talked a little bit about fitness landscapes before, that you're moving through this landscape and trying to climb, say, a fitness peak. You're running as fast as you can, but even though you're moving through the landscape, the peak is just kind of forever receding away from you. So you might be adapting, you might be changing, but you're never able to properly get up that peak. Yeah. Okay, so let's think a little bit about how we can model coevolution. I'm going to focus on host-pathogen coevolution, just because we've been looking at evolutionary epidemiology recently, and we've explored how pathogens can evolve in isolation. So where the hosts are not evolving, or vice versa, how hosts can evolve when pathogens don't evolve. So we're going to start from our basic SI model that we've had before. One can make more complex models, but the math becomes a little bit trickier. We're just going to keep things as simple as we can here, just for the sake of tractability. So we have our birth term, B of S and I. I won't define what that is yet. We have this infection term, beta SI, where beta is our transmission rate. We have a natural death rate, natural per capita death rate of D. So this is everything except for disease. And alpha is our virulence, or our disease-associated mortality rate. Again, for simplicity, we've assumed no recovery. You should all be fairly familiar with this by now. So we've recently seen how we can use these sorts of compartmental models to look at either pathogen or host evolution. Now we're going to think about what happens if they co-evolve together. Now, as before, we need to think about trade-offs, because, for example, if we're allowing host resistance to evolve, if there's no cost of resistance, then all hosts will just become resistant. It's a very trivial outcome. Likewise, if there's no cost of becoming more infective for a pathogen, it will just evolve to its maximum infectivity, right? So those are trivial examples. So we must think about some kinds of trade-offs in order to get coevolution being maintained. And so we're going to think about two different trade-offs. So first of all, for our host, we need to find its birth rate B of S and I to be equal to some other function B of U. And then the rest of our birth term is going to be the density of susceptibles times by 1 minus Qn, where this 1 minus Qn is essentially giving us logistic growth. Okay, so there's density dependence on births, where Q is telling us the strength of density dependence. Okay, so you should be familiar with this kind of formulation now. So Q here is our strength of density dependence on births. And this other function that I've introduced, B of U, is our trade-off. So our host trade-off, so some function between our level of resistance is given by this parameter U. Think of this as the strength of investment in resistance. and it's trading off with reproduction. So you generally assume that B prime of U should be negative. Things that are more resistant, if we're assuming U is your investment in resistance, things that are more resistant should have a lower birth rate. They're diverting their resources away from reproduction towards resistance. So that's one trade-off. The other trade-off, we're going to look at something a little bit different to what we've looked at before. I'm now going to assume that this transmission rate is actually a function of both our host trait U, our strength of resistance, and our pathogen infectivity V. I'll actually split this up into two functions. So beta naught is just some baseline function for, you think of this as being the maximum infectivity of a pathogen with infectivity level V. So you can think of this as being somewhat analogous to the producing lots of transmission stages, right? So where's the pathogen directing its attention? Okay. And then we're going to multiply this by function Q of U and V. So break these down. Beta naught of V is the pathogen's trade-off between infectivity. So here, this is a little bit, language becomes a little bit difficult because we end up referring to some of the same things by the same terms. It's infectivity, or let's call it something different actually. Let's call it its transmission, and we can avoid it. Let's call it between transmission. So how transmissible it is. Beta naught of V. And its specificity. Let's see if it's, there we go. We'll talk a little bit more about specificity in a moment. This other function Q is just something that's telling us our susceptibility of hosts with resistance U to pathogens with infectivity V. I'm not actually going to delve much into these functions. I just want to give an idea of how we might break these things down if we're trying to set up a system of coevolution. Okay. We're quite limited into how much we can do analytically in the kind of scope of a class here, just because the dynamics could get very complicated very quickly. So typically, a lot of these things are solved numerically. The idea here, though, is that we've got some trade-off between reproduction and some level of resistance. The pathogen experiences a trade-off between how infective it is, V, so its level of investment in infectivity and its transmission rate. And then this function here, this Q, might look a little bit strange, but it's basically saying the interactions that we'd expect between hosts with a certain level of resistance and pathogens with a certain level of infectivity. So one might set this up, for example, to assume that if V is greater than U, then the pathogen has a high level of infectivity. One could use obviously very different functions to do that. You could also have something that the pathogen has high levels of infectivity if V was equal to U. So it has to say match it. So one can then think about different types of trade-off functions as well as different types of specificity functions like this, and think about how those impact on coevolution. Okay, so that's the general setup. What I'm really going to focus though on is how do we derive some of our key results analogous to one-dimensional evolution. So just like when we had one trait evolving, we can think about our invasion fitness for a rare mutant. Okay, but now we have to think about what is that rare mutant? Well, this rare mutant could be a host or it could be a pathogen. And one of the simplifications we're going to assume is that we're still going to have this separation of ecological and evolutionary timescales. You know, the system's going to settle down to some equilibrium before we introduce a rare mutation. But we're only going to assume that there's one mutant, okay? So that mutation is either going to be in our host or in our pathogen. So that means that our invasion fitnesses for our host and for our pathogen, which are called WU and WV, are now going to be functions of 3 variables. So for our host mutant with resistance level it's invading a population that is defined by residents with traits U and V. So a resident host population that has resistance level U, and what is the pathogen's current infectivity level? V. So previously, this would have just been a function of 2 variables, but now not only is the resident population defined by whatever the host is here, it's also defined by whatever the pathogen is, because they're both evolving. Likewise, for our pathogen invasion fitness, it's defined by what is the infectivity of our rare mutant, V sub m. And this is also invading into a population that's defined by both the host that's currently there and the pathogen that's currently there. Other than that, though, we can write things down in a similar way to what we did before. So one thing to note here is that we had no recovery. And also here, there is no infected individuals who are reproducing. So that means no recovery and no reproduction from infected individuals. Infected hosts. What does this mean when we want to write down our host invasion fitness? Do we need to use the next generation method or not? No. So in other words, our I is a zombie class. It has no contribution towards fitness, okay? So we can write down our invasion fitness for hosts. In this case, it's just our per capita growth rate of a rare mutant. In our model, this is going to be B of UM times by 1 minus QN star minus beta of UMV. So this is telling us at what rate our hosts with this trait, so this level of resistance infected by pathogens that are in the resident population, times by the density of infected hosts, I star, and then minus its per capita death rate, D. So this, other than the fact that this is happens to be a function of 2 variables here, this is just like what we've done before, okay? Looking at its per capita growth rate when it's rare. Similarly for the pathogen here, IM, per capita growth rate of the IM class is going to be beta of U and VM, is now talking about the invasion fitness of a pathogen that has infectivity VM, and the hosts have resistance level U. Okay, so just to note that these 2 things are different. This gets multiplied by the density of susceptible hosts at equilibrium, minus D minus alpha. So note that these 2 things are already separate, okay? The first one is what happens if we have a host mutant, and so we would then evaluate what goes on with that host mutant. Can it invade? Allow things to settle down again to an equilibrium. Maybe this time we have a pathogen mutant, and then we look at what happens to the pathogen mutant. That's okay. So we're not looking at both of these things exactly at the same time, okay? Okay, so just like before, we can work out our selection gradients in exactly the same way. So now we're going to have 2 selection gradients. We're going to be thinking about what is the selection gradient for our host what is the selection gradient for our pathogen? So one way of thinking about this is we're in a two-dimensional space now. In fact, let's just go back a step and think about what happens if we were in a one-dimensional space. So suppose we had just one trait evolving X, 4. We would be working out what is our selection gradient at some particular point. If it's positive, then we'll increase. If it's negative, then we'll decrease. Yeah. So at any given trait value, how are we evolving? We're no longer in one dimension, we're in 2 dimensions now because we're co-evolving these traits. So with our notation here, we have a 2D space U and V. We're then going to be thinking about what is, at a given value of U and V, what is the selection gradient? Let's call this little s of U and V for our host. We also want to think about what is our selection gradient. I'll call this R of U and V for our pathogen. Okay. We're going to be thinking about 2 different selection gradients. So for our host, selection gradient, exactly the same process as before. We take the derivative, the partial derivative of invasion fitness with respect to our mutant trait, UM this time, evaluate this when UM is equal to U. Make this selection gradient as a function of both variables. So we're only taking a derivative here with respect to our host trait. We do this, all we're taking is our invasion fitness from the previous slide, and taking its partial derivative with respect to UM, and then setting UM equal to U. So you get B prime of U, so the trade-off, the derivative of this trade-off. 1 minus Qn star. And then now, this second term here, we need to take the partial derivative with respect to UM, and then set it equal to U. So you get dB to du. And this gets multiplied by I star. Do a similar thing for our pathogen selection gradient. R of U and V. So our partial derivative of pathogen invasion fitness with respect to our pathogen mutant trait, Vm. Set Vm is equal to V. So what does this look like? Well, we only actually have a Vm in this function here. So we take our partial derivative with respect to Vm. In this case, we just set it end up being equal to V. And this gets multiplied by S star. The minus D and the minus alpha just drop out from the previous slide. Okay, so we've got 2 selection gradients. Now, in one-dimensional adaptive dynamics, once we found our selection gradient, we just set one of these, our selection gradient equal to 0, to find our singular strategies, right? Now, we want to think about a pair of co-singular strategies, is when both of these are equal to 0. You can think of these, like actually, you could set this one equal to 0, and you can set this one equal to 0, and then you're looking at their intersections. In your dynamical systems parlance, right, this is then a null line when we set this equal to 0. And this would be a null line when this is equal to 0. And then we're essentially looking at an equilibrium with both of these traits, so when those null lines intersect. So if it helps to think of it in that way, that's what we're doing here. So we're seeing when these selection gradients are both equal to 0, and when they intersect, we call that point U star V star. This is a pair of co-singular strategies, because if you just had, say, U star with some V, that would give us this null line. And if we had U V star, that would give us this null line. And we want them to be both intersecting, okay? And U star V star. What does that tell us? Well, we can't descend too much from the top. We just know that these 2 terms, B prime of U times by 1 minus Qn star must equal partial derivative of our transmission function beta with respect to U times by I star. This does tell us something a little bit, though. We can kind of interpret this loosely biologically, right? This term is all about births. How many offspring are you producing? And how does that decrease as U increases? So as you get more resistant, how is your production of offspring faring? Over this is telling you here about how rapidly you're getting infected, or what is your rate of infection. So this term here is telling you, okay, as U increases, your births might be going down, but how is your rate of infection changing? So there has to be, at a co-singular strategy, a balance between these two things, right? Something about your costs of resistance versus your benefits of resistance. That's all this is essentially saying. Costs are balancing benefits at a co-singular strategy. For a pathogen, fit singular strategy equal to 0 with these equations here. Well, this tells us that d beta, sorry, I should actually add a little bit about the evaluation here, sorry. Let's just edit this equation up here. This gradient here is evaluated at U star, B star, okay? And this B prime should be at a U star. Okay, similarly for our pathogen, we're taking this derivative up here with respect to V and evaluating this at U star, B star. And gets multiplied by S star, and this must equal 0 for our second selection gradient here to be equal to 0. But S star is just our density of the susceptible population, and this can't be equal to 0, otherwise we'll end up in a trivial scenario. Our hosts are extinct, right? So this tells us that this gradient here must equal 0. S star is always positive. So that's saying that our pathogen is going to be evolving to a point at which this transmission function, which is just a two-dimensional function, must be at a turning point. Logically, we should be thinking that this should be a maximum, or at least a local maximum. It doesn't have to be a global maximum, but it should be a local maximum. We can tell a little bit in terms of sort of biological intuition just by looking at these selection gradients when they're equal to 0. Any questions so far? Yes.
Why should we know that's the local max?
Intuitively, if it was a minimum, then it would be able to move a little bit away from it and increase its transmission. So we would be expecting this selection gradient here has to evolve in the direction of this. So if this is positive, then V will increase. This is always positive. So we would always be expecting to be increasing. If you imagine just fixing U for a moment, and then thinking about what happens to V, well, V will increase when d beta dV is positive. Therefore, we're kind of climbing up a mountain. So logically, this should be a fitness maximum. We'll get onto that in just a second. Any other questions? Nope. Okay. So we can also talk about evolutionary stability in the same kind of way as we did in one dimension. Okay, so everything kind of we're doing so far is very analogous to a single trait evolving. So for our host, we're going to talk about evolutionary stability here from the perspective of each species. Okay, so from the perspective of our host, it's a co-singular strategy is evolutionarily stable if we take our second order derivative of invasion fitness and evaluate this at our singular strategy. Note that we're also evaluating our singular strategy here, our co-singular, a pair of co-singular strategies. So in our model, we have beta double prime of U star times by 1 minus Q n star minus the second partial derivative of beta with respect to U And again, this is evaluated at U star, V star. And it's multiplied by I star because that's just a coefficient here. And this has to be negative. So exactly the same logic that we've had in one dimension. We need to be at a fitness maximum. This is telling us that nearby mutants can't invade. If this is negative, then that means that we must be at a fitness maximum from the perspective of our host. Okay. For our pathogen, we do exactly the same, but just think about from our pathogen's perspective. Take our second order derivative of invasion fitness for our pathogen with respect to our pathogen trait Vm. We're now going to evaluate this again at U star V star. From above, remember this is our invasion fitness up here. We're just going to get our second order partial of theta, our transmission function with respect to V, evaluated at U star V star times by S star has to be negative. And just like before, this is going to tell us that our curvature at a co-singular strategy has to be negative. So you asked a moment ago, how do we know it's a maximum? This is how we know it's a maximum, because it follows through once we take the second order derivatives, okay? So we can never have a co-singular strategy unless our pathogen, its transmission function, is at a maximum. I'll just go back up here a moment to this slide here. Because we're only ever introducing a host mutant or a pathogen mutant, we don't actually care about any of these potential directional derivatives in other directions, right? So we don't ever end up in a scenario where we need to think about what's going on in, say, this direction here. Because we're only ever introducing a host mutant or a pathogen mutant, essentially. Pathogen, I should say. Yes.
How are we only introducing one at a time if they're coevolving?
Very good question. It's because of this separation of timescales argument. We do essentially relax those assumptions, say, when we simulate it, and you can introduce multiple at the same time. But for the purposes of the analysis, yeah, we're kind of assuming that the system approaches that ecological equilibrium, then you introduce one mutant, and then you allow things to reach equilibrium. Essentially, T goes to infinity before you introduce your next mutant. Okay, so we'll get onto this a little bit in a moment as well. Like, we can actually talk about this as a system of ODEs itself for U and V, in which case this kind of analogy breaks down a little bit. But this is kind of the setup of how we're getting there. And we're not, our evolutionary stability, we only essentially care about what is the, that we're a fitness maximum in this direction, say, or in this direction. We don't care about whether we're a fitness maximum in this direction. Yes.
Since the mutation rate of pathogens usually much faster than the mutation rate of hosts, so this assumption just failed there, I guess.
Yes. So that actually gets, hold that thought, just because we're about to get on to touching on that. So yeah, we'll come back to that in just a second. Okay, so we've talked about our selection gradients, our co-singular strategies. We've done evolutionary stability. The last ingredient is convergent stability. This turns out to be a lot trickier. One dimension is nice and simple. 2 dimensions gets a little bit nasty. The reason is, like I just alluded to, what we're really doing here in a way is we've got our ecological dynamics, which are described by a set of ODEs, but we can think of layering on another set of ODEs, which describes our evolutionary dynamics. Okay, so what we're really doing in a way is we have our host trait U, our level of resistance, is evolving over some time scale. I'll call it capital T here, so du capital T. And likewise, we have an equation for our pathogen trait here. You can also do this in 1 dimension, but in 1 dimension, the mutation rates don't matter. Okay. So what does this system essentially look like? Well, we have some functions here that describe our mutation rates for our host and pathogen populations. I call these phi of u, which is a function of perhaps the the host population density. Likewise, our mutation supply for our pathogen population is a function of number of infected hosts. And then these traits are evolving subject to our selection gradients, which we've written down. I'm writing down in its full form here. This is just S of U and V. And I'm writing them as our derivatives of our invasion fitness evaluated at U or M for reasons that will become apparent soon. So, you know, this is just a system of ODEs, right? It's a bit more complicated because we've got these selection gradients in here. But in principle, if you can solve everything analytically to put these to find these selection gradients, you can substitute these in here and you could just solve your system of ODEs like you would solve any system of ODEs. What do you know about systems of ODEs? Well, if we're in one dimension, the range of outcomes is a lot smaller than if we're in two dimensions, right? One dimension, you're just moving along a line. Two dimensions, you're in a plane now. So we can get things like oscillations, for example. Okay, so I mentioned this idea of mutation supply, and to come back to your point a moment ago, if we're just in one dimension, you know, a singular strategy will just occur when this is equal to 0. A co-singular strategy in two dimensions occurs when both of these terms are equal to 0. In one dimension, because we're talking about long-term evolutionary dynamics, this term here doesn't really matter at all, right? Because it's just telling you how long it takes to get that. We're always letting t go to infinity anyway, so it doesn't matter. Two dimensions now, well, the rates of these two things might matter, the relative rates at which this is changing versus this changing. So you asked about pathogens, they might mutate a lot faster. You might expect this term here to be much, much faster than this term here. And so this trait here might be evolving a lot quicker than this one. So we need to think about how those dynamics might mediate the outcome. In fact, I'm actually going to simplify things and even assume that they are the same. Actually, just because it shows that even when we assume a very simple scenario, our understanding of convergent stability changes in two dimensions, okay? So for simplicity, we're going to assume that actually these are constant and equal. The simplest scenario we can have. So phi of u and phi of v are just going to equal some constant phi. This is obviously a gross simplification. But we're doing this just so we can more easily derive some conditions about convergent stability. Okay, I'm going to go for a few more minutes. Like, I know we reached the 50-minute mark. I'm going to go for a few more minutes just because there's a more natural break, and then we'll have a shorter second part, okay? So bear with me, there's only a few slides to go. So like I said, this is just a standard system of two ODEs, right? Or it's a little bit more complicated than what you'd meet ordinarily, but we can do similar things. We can essentially linearize around our equilibrium. Our equilibrium here is our pair of co-singular strategies. So we're talking about an evolutionary equilibrium rather than an ecological equilibrium, okay? We can linearize around that and determine whether we, you know, a small perturbation away from that set of co-singular strategies, our evolutionary equilibrium, whether that small perturbation is going to grow or decay, just as we do when we do linear stability analysis, okay? So what do we do with linear stability analysis? Well, we need to consider the eigenvalues of a Jacobian. So we need to work out our Jacobian of these dynamics up here. Now, my simplification of taking this, setting these two things to be the same, means that we can just pull a phi out the front. Okay, that makes things a lot easier. What do we have here? Well, if we work out, in fact, I'm just going to write this out once rather than write out the full thing. we work out our partial derivatives. Because here we're evaluating our invasion fitnesses at U and V. In other words, we're taking our derivatives and then evaluating them at U, evaluating this one at V. We need to use the multivariable chain rule in order to work out what our derivatives of these things are with respect to U and V. Okay. So you can't remember this. So multivariate down here. Multivariable chain rule. So if we've got a function f of x and y, where x and y are functions of, let's call them g, functions of u and v. Let's call this one g and let's call this one h. Then if we want to take our partial derivative of f with respect to u, we take our partial derivative of f with respect to x, multiply that by the partial of x with respect to u, and then add on our partial of f with respect to y, multiplied by the partial of y with respect to u. Okay. So if we apply that, then we end up with something that looks kind of complicated. We end up with our second order derivative of host invasion fitness, WU, with respect to U. With respect to UM. And then we have a cross derivative with respect to UM and U. We have something that looks very similar for our pathogen. Our second order partial derivative of pathogen invasion fitness with respect to our pathogen mutant trait to VM. And we have its cross derivative with respect to VM and V. And then in the minor diagonal terms in the top right, we end up with second order partial of WU, so host invasion fitness with respect to UM and V. And the bottom left, we have our second order partial of invasion fitness for our pathogen with respect to VM and U. So this is kind of messy, but really the thing to remember here is all we're doing is we're doing our standard linear stability analysis around an evolutionary equilibrium. So we need to think about our Jacobian of our ODEs from this slide up here. To get that, we need to use the multivariable chain rule. But ultimately, the eigenvalues of this matrix will tell us whether our evolutionary equilibrium is locally asymptotically stable. In other words, is it convergent stable or not? When we evaluate this, U star, B star. So I've not included all the evaluations above just because it's kind of messy. Okay, so some of these should look a little bit familiar. Some of these derivatives. As with any 2x2 matrix, if we want to find out the signs of the signs of its eigenvalues, or at least the signs of its real parts of those eigenvalues, we can just look at the trace and the determinant and use the Routh-Hurwitz conditions. Okay. So let's think about what happens to the trace here. So the trace of this Jacobian being less than 0, what does that imply? Well, what are the elements on the main diagonal? This element here and this element here. So we add these two things together to get the trace. We evaluate them at U star, V star. And this has to be negative. Does anyone recognize these things here? I'll forgive you if you don't recognize. Yeah. Yes. So these are convergent stability in one dimension. So can you conclude anything about at least the trace here based on the convergent stability of these things in isolation? So in other words, convergent stability of our host trait in one dimension, convergent stability of our pathogen trait in one dimension. Can we conclude anything about the sign of our trace here?
If they are both convergent stable, then we can.
Then at least
this is.
Yeah, exactly. So this can be negative. Well, this will be negative if they are both convergent stable. Okay, so at least that's kind of one thing. However, they don't have to both be convergent stable, right? If this one has a much larger magnitude than this one, say this is actually convergence unstable, this one's convergent stable, this could still be negative. So it tells you something about convergent stability in one dimension doesn't necessarily carry over to convergent stability in two dimensions. So this tells us that at least one, but not necessarily both, must be convergent stable when we just look at them in isolation, in order to get convergent stability when they're coevolving. But we also need our determinant to be positive in order for this Jacobian to have eigenvalues with negative real parts. So this is only one piece of the puzzle. I'm not going to work out the determinant of this, but we can see that even if this is satisfied, it's possible, depending on what these functions look like here, that our determinant is negative. So this tells us that convergent stability in one dimension is neither necessary nor sufficient to give us convergent stability in two dimensions. The only thing we can conclude is that at least one of these has to be convergent stable when it's viewed by itself. So even if our trace is negative, our determinant may also be negative. This is all we're really saying is that once we go from evolution to coevolution, things get a lot more complicated, and you can't necessarily just carry over what you learned from one-dimensional from the one-dimensional systems to the two-dimensional systems. Note that this convergent stability is very different to what happened with evolutionary stability up here, because we're essentially just looking here in a line. You know, we're thinking just about what's going on with the mutant trait, or what's just going on in our host or mutant trait with our pathogen. We're not having to account for these cross derivatives, and therefore we're not having to account for the mutation rates and things like that. It's essentially just looking at the curvature. So these things are directly analogous to what we had in one dimension. Whereas our convergent stability becomes a lot trickier. It becomes even more trickier if we think about different mutation rates. If we had different mutation rates in here, this is going to complicate this even more. So if you don't know anything about the mutation rates, it's very difficult then to understand what's going on with convergent stability. So that's definitely a challenge in this area of modeling. So people often just kind of make this simplifying assumption in order to get some convergence. There are other things that we can do. Essentially, we can talk about conditions which I'm not going to go into about strong convergent stability or absolute convergent stability. So strong convergent stability is basically saying that every single path in this two-dimensional trait space converges towards U and V. In other words, if this is my U star, V star, we can draw a ball around it where every single trajectory converges. But to go back to your point earlier, we might not necessarily care about every trajectory because many of these trajectories might not be very realistic. Say pathogens evolve much faster than hosts. We don't necessarily want it to converge for every possible combination of mutation rates. Actually, I did some research recently with one of my PhD students showing that you can actually make some simplifications in that kind of limit where pathogens evolve much faster than hosts. Okay, we're nearly done for this section. So because of this, often we end up solving these systems numerically, but we can get a real rich set of outcomes. These are actually some simulations. So just like those evolutionary trajectories that we looked at before, these are coevolutionary trajectories. And we can see a much richer set of outcomes when species co-evolve than when they just evolve by themselves. So this one on the left here, this is host resistance, and this is pathogen infectivity going through time. So these two on the left are one scenario, and the two on the right are another scenario. What's going on here is we're getting this cascading branching. So first, the host population branches into two levels of resistance. This actually creates a feedback loop, which means that now the pathogen can branch into two different levels of infectivity. That actually creates a feedback loop, which means the host can now branch into another level of resistance. The pathogen then can branch into another level of infectivity, and so on. So we can get this cascading effect where if we just had one of these species evolving, maybe you might get just one branching event, perhaps. When they're co-evolving, they're essentially creating additional niches. So this feedback loop between them can generate diversity and lead to this runaway process. The other kind of interesting thing on this right-hand side here is we're getting, say, resistance initially increases, but then it gets to a point where it's too costly. And so now we get selection against resistance. Here, infectivity basically tracks resistance. And this process can go on indefinitely with resistance increasing, then infectivity increasing, resistance decreasing, then infectivity decreasing, and then starting again. A little bit like if you're familiar with Lotka-Volterra models, predator and prey, a cycling between prey and predator in ecological sense, with our predator tracking our prey density. You get a similar thing here with our pathogen trait tracking our host trait. Yes.
So again, after these branching points, our analysis doesn't work anymore.
Yeah, here basically now you'd have to be thinking about 2 residents or 3 residents or 4 residents, and so inevitably it becomes intractable. Yeah. Okay, guys, that's the end of the first part. So let's have a little break for, let's say, come back at 22, and then we'll go through the final section. Okay. Okay, so we've done the more complicated version of coevolution, more complicated because we're actually accounting for these ecological dynamics. We're going to finish with something a bit simpler, but something quite cool, and taking us all the way back essentially to the start of the course, where we talked about population genetics. So we're going to talk about coevolution now in a population genetics framework. And we're going to be stripping out all of our ecological dynamics. So here we're just being concerned with frequency-dependent dynamics, so frequency of alleles. We're assuming our populations are infinite, they're asexual, they're haploid, and there's just one genetic locus in each of our hosts and our pathogens. There are 2 alleles, A and B. Okay, so about the simplest coevolution model we can construct in population genetics. We're going to see if this gives us any interesting insight. I said here we shall first assume. I'm going to get time to write the rest of my notes, which is a good thing because we don't have a lot of time left in the lecture. We're going to assume that pathogens can only infect hosts with a matching allele. It's actually known as a matching alleles model. Surprise, surprise. And this is the only difference in phenotype caused by these alleles. So otherwise, everything's the same. We would have the same intrinsic growth rates and the same intrinsic transmissibility rates and all of these sorts of things. So the only difference is what they can infect, or from a host perspective, what they can resist. So we can talk about this in terms of something called a specificity matrix. This is actually a discrete version of our function Q that we had before, right? So this was some continuous function of resistance and infectivity. We can kind of do a discretized version of this, which is a bit easier to look at. So we have a specificity matrix where we just write down our alleles in our host and our pathogen, so 2 alleles in each of them. And this specificity matrix, I'm going to say this is essentially the probability of infection if they come into contact with each other. So given that pathogens can only infect hosts with a matching allele, pathogen A has probability 1 of infecting host A. Pathogen B has probability 0 of infecting host A, and vice versa. Pathogen A has probability 0 of infecting host B, and probability 1. Nice matrix. Daniel, this should look very familiar to you. There we go. And Barbara, actually, this should also look familiar to you. Okay, so we're gonna, there are different matrices that we can look at, by the way. This is just as I talked earlier about how there are different functions of this Q and UV that can maybe tell you different things about the underlying mechanisms involved in host pathogen interactions. For example, we could have a matrix that looks, I'll just draw it here like this. We could have something that looks like this. For example, we could have different numbers in here. This tells you here that pathogen B can infect both hosts, but pathogen A can only infect one of these hosts. Maybe there's a cost, another cost associated elsewhere. It can infect both hosts if it's a generalist, but maybe it can infect them both less well. This one here is a specialist and can infect it well. There are different assumptions essentially that we can make and look at these different specificity matrices and see what impact they have on coevolutionary dynamics. We're going to focus on this very simple one here. This actually has sort of grounding in things called non-self recognition systems. So, you have sort of proteins on your cells that basically say, I'm self, this belongs to me. And we have different proteins on our cells. And your immune system will attack things that are non-self, that don't have the right. You can think of this as being like a lock and a key. If the pathogen has the right key, it can get bypass immunity and infect the host cell. If it doesn't have the right key, then it's going to get killed. This kind of non-self recognition is as well why if you have, say, like an organ transplant, you need to take immunosuppressive drugs to stop your body from rejecting the organ because it's seen as non-self. There's also features in infectious diseases. Okay, so what we're going to do is we're going to define H and P to be the frequencies of allele A in our host and pathogen populations, respectively. So H is the frequency of allele A in the host. P is the frequency of allele A in the pathogen. And we're going to follow replicated dynamics. We talked about replicated dynamics when we first started going into adaptive dynamics. We can apply them in population genetics as well. Okay, so it's essentially the same equations that we've seen before. The frequency of allele A in the host population is going to change according to dH dt. Using our replicated dynamics, this is going to be H, our frequency, our current frequency. And then here we have our per capita growth rate, or in this case, they'll end up being the same as the fitness of allele A minus the mean growth rate of the host population. And similarly for the pathogen population, change in frequency of allele A is going to be dP dt, which is P, frequency of allele A at the moment. That's by the per capita growth rate, in this case, fitness ends up being the same thing, of allele A minus the mean of the population. Okay, so these are just a couple of ODEs. System of nonlinear ODEs, we can solve those, right? So that's all we're doing here. Solving a system of nonlinear ODEs. So for simplicity, I'm going to define, say that the per capita growth rate, like I say, essentially this is the same as the fitness here, of our A allele in the host population is going to be 1 minus P. Why would that be the case? Well, this is basically saying these hosts here with allele A are going to be fittest when none of the pathogens have allele A. Right? The more pathogens with allele A around, the lower their fitness is going to be. And that's because pathogen A can infect host A. Right? For our B allele, it's just going to be the opposite of that. P. This is the frequency of allele A. The more allele A is around, the fitter B is going to be. And then for our pathogen, I'm going to switch these around just because I've got them around the other way in my notes. Don't get confused. For our pathogen, allele A, it's just going to be have fitness H. So pathogens with allele A are going to have fitness that is equal to the frequency of hosts with allele A. And again, that's because these pathogens are going to be fitter when there are more hosts that it can match. And finally, pathogens with allele B are going to have fitness 1 minus H. This 1 minus H here is just the frequency of hosts with allele B. Yeah.
Why would we need the specificity matrix when we can define it? Like this seems simpler than the matrix that we have up there.
Yes, this informs the next step below. If I'd used the other gene for gene on the 1101, then it would give me a different matrix below. This is just a way of representing the information in differently, if you like. Or rather, this is one particular way of setting up these, I'm defining these fitnesses here. There are many different ways in which I could define these fitnesses, or per capita growth rates here, because they're the same thing, coming from the same matrix. For example, I could say that all of these were like e to the minus something, right? So there are many different ways in which one could do that, but would all still represent this same specificity matrix here. Can anyone tell me what this is in a sense of a certain type of selection that we met really early in the course?
Negative frequency dependent.
Yes, I was only looking for frequency dependent selection, but frequency dependent selection is the key point. Yeah. So from the host perspective, it is negative frequency dependent selection. So yeah, this is frequency dependent selection, but now rather than it being the frequency of your own allele, or your frequency of alleles in your own population, it depends on the frequency in the others. So it's actually indirect frequency dependent selection. In a sense, because of the feedback that occurs between them, as alleles change in the other population, that affects alleles in your current population, which feeds back and affects alleles in the other population. So you end up with this cycle going here, this loop. Okay, let's do a little bit of analysis. What is the mean per capita growth rate of each species? I'll do the host one with you here. So the mean for the host, well, we just take a weighted sum. What is the per capita growth rate? Except per capita growth rates and fitness here are essentially the same thing. Of allele A times by the frequency of allele A, which is H. And I take the per capita growth rate of allele B in the host population, times it by the frequency of allele B, which is 1 minus H. I've just defined what these are above. So here we're going to have 1 minus P times by H, added to P times by 1 minus H, which is just H plus P minus 2HP. Have a quick go at doing the same for the pathogen population, just to make sure you've understood what I've done there. Taking a weighted sum over the fitnesses or per capita growth rates here of our pathogen population and their frequencies. Okay, so we do exactly the same as we've done for our host population. Take the per capita growth rate of individuals with allele A, multiply by their frequency, which can be P. Similarly for allele B. So we're going to have H times by P plus 1 minus H times by 1 minus P, which is just 2HP minus H minus P plus 1. Okay, nice simple weighted sum. So now we've got all of the ingredients, we can rewrite down our ODEs from above. So for our host, we had H, that's by MH of A minus MH bar. Note that we've got a system actually here of 4 ODEs technically. But because we're dealing with frequencies, we can always work out the frequency of allele B if we know the frequency of allele A. So we can reduce it from 4 to 2. Okay, so substitute in our values for MHA and MH bar. This is just a simple substitution. So what's our MHA? It's going to be 1 minus P minus H. Yeah, so sorry, our MH of A is 1 minus P, and then we're subtracting H plus P minus 2HP. So we end up with all in all 1 minus P minus H minus P plus 2HP. Which just simplifies down to being H times by 1 minus H minus 2P. That's by 1 minus H. Exactly the same for the pathogen. Substitute in pathogen fitness, so pathogens per capita growth rate. In this case, it's going to be H for allele A. And then subtract the mean fitness of the population, or mean per capita growth rate of the population, of minus 2HP plus H plus P minus 1. Simplifies down to being P times by brackets 2H brackets 1 minus P plus P minus 1. Like I said, a lot of this ultimately just boils down, even though we're modeling evolution, we can apply the skills we've learned from any system of nonlinear ODEs. So a system of 2 ODEs with 2 variables. So just to check you guys remember how to do this, I need to find how many equilibria this system has and what they are. Okay. Okay, let's work through this. Okay, so for us to have an equilibrium, both dHdt and dPdt have to be equal to 0. So if dHdt is equal to 0, well, from this line up here, we know that either H has to equal 0, or H has to equal 1, or P has to equal a half. Right? Just because one of these factors has to be, at least has to be equal to 0. So either H has to equal 0, H has to equal 1, or P has to equal a half. And if we look at dP dt being equal to 0, well, by the same logic, either P has to equal 0, or P has to equal 1, or H has to equal a half. So now we can think about which combinations of these work in order to work out how many equilibria we have. So if H is equal to 0 here, this bracket here of 2H minus 1 can't be 0. That means that either P has to equal 0 or P has to equal 1. So that means that H is 0 and P is 0, or H is 0 and P is 1 are both equilibria. Think about what happens if H is equal to 1. So if H is equal to 1, exactly the same thing. This 2H minus 1 can't be equal to 0, so either P has to equal 0 or P has to equal 1. So if H is equal to 1, then we know that either P has to equal 0 or P has to equal 1. And finally here, if P is equal to a half, this is equal to 0. P is equal to a half here, 1 minus P is equal to a half. So this term in brackets here has to be equal to 0, so 2H minus 1 must equal 0. So P is equal to a half, H must equal a half. We have 5 equilibria. Now, show of hands, who got 5? Yeah. Okay. So 5 equilibria. So the next step is to look at our Jacobian to analyze the stability of these equilibria, okay? So our Jacobian, I'm just going to write this down rather than deriving it because it's a relatively straightforward one to do. We have 1 minus 2h multiplied by 1 minus 2p in the top left, minus 2h times by 1 minus h in the top right, 2p times by 1 minus p in the bottom left, and minus 1 minus 2h times by 1 minus 2p in the bottom right. Now, if you evaluate any of the, well, actually, I'm going to take a quick step back here. Just because I heard a couple of people mention it. Are any of these trivial equilibria? I'm seeing some nods and some shakes of heads. Someone, I'm going to pick on you, Javad, just because you were shaking your head. Were you going for they are trivial or non-trivial?
0, 0.
Yeah.
It means that there is only one allele, but there's no evolution occurred yet. So,
well, you could have evolved to that point, right? It means that there's no more variation in there, right?
yes.
So you're right. This is something that tricks people or catches people out, I should say. We're so used to modeling population densities, and we've been doing this extensively. Your brain is probably trained to seeing 0, 0 as trivial. We're modeling frequencies here. Frequency of an allele, frequency of an allele. So 0, 0 is basically, well, actually all of these for here are states where one allele has fixed in each of the populations, right? So one of the alleles is at 100% frequency, the other allele is at 0% frequency. So here, allele A in the host population is at 0% frequency, and allele A in the pathogen is at 0. Here, allele A is at 0 again, but allele B is now at 0. So allele A is at 100% and so on, okay? So that's what I talk about here when I say about boundary equilibria, is that you can think of these as being, because these are frequencies, you can think of this as being a square in the 2D plane, a unit square, and we can't go outside that unit square, right? Because the maximum any of these things can have is a value of 1. We can't have a frequency above 1, and they can't have a frequency lower than 0. So everything is contained within this unit square. So when I talk about a boundary, I'm talking about something on that boundary of that square. And if you evaluate any of these for equilibria here, you always end up with one of your eigenvalues is equal to 1, one of your eigenvalues is equal to -1. Can anyone remember from systems of 2 ODEs what this means in terms of stability of these different equilibria on the boundaries? I've heard of you, saddle?
Yeah.
There we go. So if one of our eigenvalues is positive and one of our eigenvalues is negative, that means we have a saddle. What that really means is that you're being drawn towards this equilibrium from one direction and you're being pushed away from it in another direction. So we have a saddle on boundaries. What is really going on here is that if you're on one of those boundaries, say you're at a point 0, 1/2, that means that one of these alleles is fixed. And we don't actually have any mutation in this model. So there's no way of generating any variation again. So all that happens is you start somewhere along that boundary, say 0, 1/2, you'll tend to either 0, 0 or 0, 1. And then you stop. But if you come off that boundary, you're going to get pushed away from it. You're not going to get pulled back to that boundary. Okay, because it's a saddle. I realize I cannot spell boundaries, apparently. Yes.
I still don't quite understand why 0, 0 isn't trivial?
So when we're talking about trivial equilibria, usually in dynamical systems, we're talking in the population dynamics context, right? We're talking about things that are extinct. But here we're talking about frequencies of alleles. So 0, 0 is no more trivial than 1, 1. Because here this is just the frequency of allele A in each of our populations. So this 0, 0 is just saying allele A is extinct in both populations. So there's only allele B. So they're trivial in the sense of, as Javad was saying earlier, they're trivial in the sense that there's only one allele in each population because there's been fixation. But they're not trivial in the sense of what we would usually talk about trivial systems where the populations are extinct, so nothing can happen. Because at this point, we don't actually know anything about their stability. You could be oscillating around them, for example, in which case you oscillating like towards them, away from them, whatever, right? Some weird dynamics. Okay. So trivial usually we refer to just as being something extinct. Okay, so let's think about our internal equilibrium. So I'll talk about this internal or interior equilibrium. That's a half. So if we evaluate our Jacobian from above, I'll call this J star. The elements on the main diagonal go to 0. The top right, we have minus a half, and the bottom left, we have a half. So what I've done here is I've just taken this Jacobian, evaluated H and P at a half. Should be pretty clear to see that when H is a half and P is a half, this goes to 0. So just this, and these end up being non-zero. Okay, you can work out your eigenvalues directly, but let's just consider the trace and determinant. So our trace, which I'll call tau, is going to equal what? 0. And I'll call delta my determinant is going to equal what? 1/4, which is positive. If I look at my discriminant, so my trace squared minus 4 times by my determinant, remember this is going to tell me if my eigenvalues are real or complex. I'm going to have 0 squared, which is 0, minus 4 times by 1/4, which is minus 1, which is negative. So my eigenvalues are complex. In fact, they're imaginary. They have 0 real part. One could also see this if you just tried to solve the characteristic equation of this directly, you would end up with lambda squared plus lambda squared plus 1/4 is equal to 0. Okay, so our eigenvalues are imaginary. Can anyone remember what that means in terms of dynamical systems?
Circles in this case.
Yeah, so they're neutrally stable. So what I'm going to do is actually I'm going to sketch a phase plane of this. It's easier to see what's going on. So this is my frequency, the host on the horizontal axis, pathogen on the vertical axis. Remember, I've said that we're bound to this unit square. I'm actually going to sketch on my null lines. So my null lines are at H is 0, H is a half, and H is 1. P is 0, P is a half, and P is 1. So we actually have null lines on the axes here as well. It's kind of hard to see them. And then we have equilibria on our boundaries. And at this interior equilibrium in the middle. Okay, so if you can work out the direction field. I'm not going to do this just because time is limited. If you work out the direction field, say on each of your null lines, you'll end up with an arrow going down here, one going right here, one going up here, one going left here. This should make some sense. Because the pathogen wants to match the host always, if the host has its allele A as at frequency 0, in other words, it's only allele B, then the pathogen is going to want its frequency of allele A to go to 0 as well. Likewise, if the host has, if the pathogen has fixed allele A, if the pathogen has fixed allele B, in other words, P is equal to 0, the host is going to want to get away from it. So it's going to want to move away. Okay. Because these are continuous, these arrows will carry on into the middle. So as we're crossing these null lines here, have similar dynamics. And I'm usually terrible at actually sketching these things. If we then draw some orbits, we'll end up with these closed orbits that look like this, but our system is neutrally stable. These are neutrally stable cycles rather than a limit cycle. What that means really is that if you get pushed off one of these orbits, or if you start with a different initial condition, you orbit around based on that initial condition or based on that perturbation. You don't get pulled back to just one stable orbit. So these allele frequencies, therefore, are just going to keep oscillating, right? And this harkens back to that, again, idea of like a predator-prey system. This gives you essentially what you see in a classic Lotka-Volterra predator-prey model, but in an evolutionary sense. So here, what's going on? Well, suppose allele A is at a low frequency in the host population, the pathogen population as well. So we're starting off close to the origin. And then there'll be selection for the host to move away from the dominant pathogen allele, which is B. So A is going to increase in frequency. But then as A becomes more frequent, more common in the host population, pathogen is going to be switching towards allele A. Now the host switches towards, then we end up here. Now the host switches towards allele B. H goes towards 0, and then the pathogen tracks it. So the pathogen is just kind of following it all the time, if you like. And these allele frequencies will then oscillate indefinitely. Yes.
So one is that doesn't this like violate our separation of time scales assumption? Because we're assuming that one mutation in either host or pathogen happens and it comes to fixation.
Let me stop you there. So we're not dealing with adaptive dynamics now. So we've moved back to population genetics. So adaptive dynamics is all about this separation of ecological and evolutionary timescales. Here we're just dealing with population genetics. So we're not. I think we've gone basically all the way back to the beginning of the course where we were dealing with population genetics. So there's no assumptions here about separation of timescales anymore.
I mean, we can't describe this phenomenon with adaptive dynamics if we make that assumption.
I mean, we could end up with a model that gives us cycling something like this. What would be, so this is a cycle just like the cycles that we've shown before. So you can end up with cycling. It's cycling again in an evolutionary sense, like the ecological dynamics die down, and then you would introduce a mutation, and then there's no guarantee that that mutation is, say, always going to be increasing. It could be going the other direction, be selected for. So the direction of selection essentially shifts. What's going on here? Like you think about like your direction of your selection gradient in one dimension when you do it in adaptive dynamics. At any given value of whatever your trait is that's evolving, it's always positive or negative or 0. But in 2 dimensions, whether it's positive, negative, or 0 is going to depend on the other trait as well. Yeah. Any other questions?
I think that's
okay. Okay, so I just wanted to finish here on this slide here. This is like an illustration of what's going on here. So we've got, I call them type A and type B. These are your allele A and allele Bs. This is what's going on essentially. Infection by matching parasites drives selection for a different host type. That's what we're seeing above. So these green creatures, you can think of as being the ones with allele A, and these pink dots as being parasites or pathogens with allele A. So now these hosts, say, have allele B. Pathogen is still allele A. More common host type drives selection for a matching parasite or pathogen. So now these stars here are the ones that can infect these hosts. This is allele B in our pathogen. So we have allele A in our host, allele B in the host, and allele B in the pathogen. And then infection again by matching parasites or pathogens drives selection for a different host type. So again, this is the power of coevolution, that these kinds of non-equilibrium dynamics, these cycles could persist indefinitely, because there's the pathogen is essentially just always tracking its host. So as soon as, this goes back all the way to the idea of the Red Queen, you can find her. They can never escape. The pathogen maybe, or the host maybe gets a little bit ahead, pathogen catches up. You kind of reset the scenario again. Maybe host escapes, pathogen catches up, resets the scenario again. Okay, that is it for today. So like I said, we've moved beyond this idea of one-sided adaptation to think about coevolution in two species. The analysis is typically just a lot more difficult, as we've seen, especially when it comes to things like convergent stability, because you need to think about relative mutation rates. But we can also look at like population genetics models like we've done now, and get a bit of insights as to what goes on with these coevolutionary dynamics. I will also say we've really barely scratched the surface of coevolution. This is one lecture. You could spend an entire course talking about it. But hopefully this has given you an idea that some of the things that we've done in just one dimension can be extended to think about more interesting and realistic biological scenarios. With that, have a great weekend. Remember, there is no lecture on Monday. Project's due next Wednesday. Have a great weekend. I'll see you next week for the final course review.